Welcome everyone to the Critical Issues webinar series. My name is Cassandra Rose and I'll be moderating today's Critical Issues webinar, Ocean Acidification Impacts on Fisheries. We'd like to thank the co-sponsors of our webinar today, the Consortium for Ocean Leadership, NOAA Ocean Acidification Program, and Northeast Coastal Acidification Network. In today's webinar, we'll be having 3 speakers discussing ocean acidification impacts on fisheries. First, Bärbel Hönisch from Lamont-Doherty Earth Observatory at Columbia University will discuss the geological record of ocean acidification. Next, we'll hear from John Hare from NOAA Fisheries Narragansett, who will discuss ocean acidification impacts on fisheries. We'll then hear from Hauke Kite-Powell from Woods Hole Oceanographic Institution, who will talk about implications of ocean acidification for shellfish. The presentations will be followed by a panel discussion where questions from the audience will be addressed by the speakers. And now we'll turn the slides over to Bärbel.
Good morning, Cassie, and thank you everybody for tuning in. So I was asked to talk about the geological record of ocean acidification to give a little bit of a perspective over what we're doing today with our increasing carbon dioxide levels in the atmosphere. But before I do that, I wanted to give an introduction into what ocean acidification really means. So we all know that the levels of atmospheric carbon dioxide in the atmosphere are increasing, and you have all heard about the problem of atmospheric carbon dioxide and global warming. But in addition to global warming, the carbon dioxide in the atmosphere will also dissolve in seawater. And you can see that over here on the screen on the left-hand side. The carbon dioxide will not only dissolve in the water, but it will actually react with water, and it will form aqueous carbon dioxide, and that will then dissociate into carbonic acid, bicarbonate ion, and carbonate ion. And so while organisms in the ocean are interested in the presence of carbonate ion and bicarbonate ion for secreting their shells, the increased abundance of carbonic acid actually forms a problem. And it turns out that the more carbon dioxide we dissolve in the water, we're actually taking away the carbonate ion concentration in the water and will turn it more and more into carbonic acid. And that is a problem in the ocean. And so when we're looking at this, we're really interested in the hydrogen ion concentration in seawater, which will increase when we have more carbonic acid in the water, and that will make the water a little bit more acidic. And what that means is that with the increased hydrogen ion concentration, that is measured as the negative decadic logarithm that tells us the pH. So when the acidification increases, when we have more hydrogen ions, the pH will actually go down. So now the organisms, as I mentioned before, they are indicated in, they're interested in the presence of carbonate ion in seawater. These are pictures of planktic snails that have been grown under controlled conditions in the laboratory. On the right-hand side, that is a shell that is grown under normal modern conditions. And then the further left you go, it shows shells that were grown under acidification conditions that we expect for the end of this century. And the more time they spent in that water, the more dissolved these shells became and the more malformed they became. And these are specifically planktic snails, and you may have never heard about them or never looked at them. They're actually very pretty. But these organisms are important because they're important parts of the food chain. So for instance, these snails are often eaten by Pacific salmon, and so if you imagine that these shells now have, or these snails now have thinner shells, they're more vulnerable to predation by all kinds of organisms, and therefore they might disappear in the future, and that might take away some of the livelihood of the Pacific salmon. To put this all into perspective, this figure over here shows you a timeline. On the top you can see from 1900 to the year 2300. And we see 3 panels. The top panel is carbon dioxide, the middle panel is surface temperature of the ocean, and at the bottom panel you can see the sea surface pH. And the red lines and the blue lines, they indicate what we call representative concentration pathways that were used by the Intergovernmental Panel of Climate Change. And it looks at concentration pathways or emission scenarios until the year 2100, and these values 8.5 and 2.6, they actually relate to what kind of warming we might have at that time, or what the radiative forcing would be. These are not numbers in temperature. But you can see that the carbon dioxide from the year 1900 until the year 2000 has actually already increased by about 40%. And by the year 2100, if we follow the most extreme concentration pathway, then it might go up to somewhere near 1,000 parts per million. If we stay at modern levels, then it will still be 40% higher than what we have seen in the in pre-industrial times. Now, when you look at the middle panel, you can see the associated warming with that, so the well-known problem of global warming. On the bottom panel, you can see the pH changes. And so from the year 1900 on the left-hand side to the modern, we have already dropped pH by about 0.1 pH units. And with the most extreme concentration scenario, we might drop that by another 0.3 to 0.4 pH units. 0.1 to 0.3 may not sound very much, but because this is on a logarithmic scale, this actually means quite a lot in terms of acidification. So we're really talking about acidification or increase of hydrogen ion concentrations by about 30% until today, and this might actually increase, or it might double by the end of this century, and that is a big problem for marine organisms. And so that is actually shown on this slide. On the right-hand side, you have the modern conditions, or the more basic seawater conditions. On the left-hand side, in red, you see the more acidic conditions that we're expecting for the end of the century. And these are different pictures of organisms that have been grown under these different conditions. I've already shown you the snails on the bottom. On the right-hand side, you can see a tile that was put into a coral reef under modern pH conditions, and you can see it's very colorful, so little corals have grown on that. If you put the same tile into conditions that we're expecting for the end of the century, then you see that it's mostly overgrown by algae, so the corals cannot grow under those conditions. In the middle panel, we can see coccolithophores. These are planktic calcareous organisms that, again, form part of the bottom of the food chain. And on the right-hand side, you see the pristine shells that were grown under normal conditions. On the left-hand side, you see the more malformed when they're growing under conditions that we're expecting for the end of this century. Similar on the left-hand side, there's an actual snail that is pitted that is not that has not the same shell that it would have under normal conditions. So many of these organisms you may not care about, but as I mentioned before, they do form the bottom of the food chain, and so therefore they can have repercussions into our food system. So as I mentioned in the beginning, I'm not actually working on these modern acidification problems. This is what the following two speakers will be talking about, but I wanted to look into the geological record of ocean acidification. And what you can see on this slide is a 300 million year record of diversity of different forms of calcifying organisms in the oceans. At the bottom we see shallow reef builders like corals, and we have some foraminifers. Those are organisms that we use a lot in paleoceanography, and then again some of those calcareous nanofossils like the coccolithophores that I showed you in the previous slide. The reason why we're doing this is because in the previous slide I showed you all kinds of organisms that are affected by acidification, but it actually turns out that not all organisms are affected in that way. And so it makes it very difficult for us to predict where the ecosystems are going in the future. By looking into the fossil record, we can look at what happened to these organisms in the past when they may have been affected by ocean acidification. And these diversity trajectories, so that means the blue lines and the black lines, they indicate by thickness how many species there were present at any given time in Earth history. And so the thicker the line is, the more species there were. The thinner the line is, the less species there were. And so the vertical red lines over here, they indicate actual mass extinction events, starting 250 million years ago with the Great Dying at the end Permian, 200 million years ago at the end Triassic, and we had a couple of oceanic and oxic events, the well-known extinction of the dinosaurs that happened with the asteroid impact at the end of the Cretaceous, the Paleocene, Eocene thermal maximum 55 million years ago, of which we don't really know what caused it, and then the last deglaciation when a lot of carbon escaped from the ocean and acidified the surface ocean. And so these little pictures that you can see on top of this, they indicate the potential causes of acidification, like massive volcanism at the end Triassic and Permian, the asteroid impact. At the PETM, there may have been methane hydrate release, or there may have been permafrost thawing and other events. But you can see that many of them actually made those diversity trajectories thinner, meaning that there was extinction at that time. And that is the reason why ocean acidification has been considered as a potential cause for these extinction events. So we wanted to understand whether that is really true and whether we can say that acidification really caused these extinctions. But in order to do that, we cannot hold a pH meter into the ocean because it will not tell us what the pH was 300 million years ago. So what we're doing in geochemistry is that we are actually using proxies. And proxies are so-called stand-ins or surrogates for environmental parameters that can no longer be measured directly. And to compare that to what we're doing today, or to give an example of what a proxy might be, is if you imagine you don't have a thermometer and you're trying to find out how warm it is outside, you would look out of the window and infer from what other people are wearing whether it is warm or whether it is cold. Now our proxies, our geochemical proxies, are a little bit more sophisticated than that, and I do not have the time to go into all the details, but what I can tell you is if we have higher carbon dioxide concentrations in the atmosphere, so if you look at the red values on the left of this screen, higher carbon dioxide in the atmosphere means that we will dissolve more carbon dioxide in the water. That means the pH will drop, and this drop in pH will actually be recorded in the boron isotopic composition of planktic organisms like these foraminifer shells that you can see over here. The boron isotopic composition is given in delta 11b. This value will decrease when the pH is low, and it will increase when the pH is higher. So on the right-hand side is a condition where the carbon dioxide in the atmosphere is low. That means less carbon dioxide in the water, higher pH, and therefore higher boron isotopes. Now these plankton shells at the end of their life cycle will sink to the bottom of the ocean. They will litter the seafloor. We can take sediment cores out of that seafloor, then pick those fossil foraminifer shells out of it, analyze their boron isotopic composition, and then infer what the pH and the carbon dioxide of the seawater was at the time, and therefore we can say something about the carbon dioxide in the atmosphere. To give you an example of how this might work, we can look at the last 400,000 years. So these wiggly lines over there give you several different records of carbon dioxide in the atmosphere that was measured in ice cores. So the pictures on the top left show you ice core pictures, and then this is a slice of an ice core that shows you the gas bubbles that were trapped in that ice that contain old ancient air. And so you can see that over the last 400,000 years, the carbon dioxide levels were going up and down about every 100,000 years. Before the industrialization, the carbon dioxide levels were at 280 parts per million. We have now increased that to about 400 parts per million. But what we're interested in is what is the acidification of the surface ocean as we're going from an ice age about 20,000 years ago, for example, into the pre-industrial. And so the picture on the top right shows you a map of the oceans that actually shows you a pH map, but what you really only care about is that little dot over there that indicates ODP 668. That is a sediment core that I have analyzed for this. And I analyzed these planktic foraminifer shells that are shown below. And you can look at the pH that we inferred from the boron isotopes that indicate that the pH during the last glacial was about 0.15 pH units higher compared to the pre-industrial. So from all kinds of evidence and other proxies, we can say that during the deglaciation, that means from the transition from the ice age 20,000 years ago to the pre-industrial, the surface ocean warmed by about 2 to 3 degrees Celsius. The pH dropped by about 0.15 pH units. And we did see calcification changes in these planktic foraminifers. They actually grow thinner shells today compared to what they grew in during the ice ages. But what is important is that for this transition to happen, it took about 6,000 years. And so if we try to compare the pH change at that time to the pH change that we're expecting over the current century, then the pH change at the time was about 0.002 units per century. And remember what I said in the beginning, the current pH change is somewhere on the order of 0.1 to 0.4 units per century. So what we're doing today is much, much larger than what happened at the end of the last deglaciation. And so therefore we went to a much larger event, and that is the Paleocene-Eocene thermal maximum. That was an event that happened about 56 million years ago. And what you can see here is a map of sediment cores that have been studied for this. It was a global event that has been found everywhere in the ocean and also on land. These are some records that I have worked on. But what is important about this event is if you look at the figures below, they show sediment cores. And these sediment cores were taken at this location of Namibia in the South Atlantic, where you can see ODP 1263. That's one of those cores. And what you can see is that these sediment cores actually change color from the bottom to the top. And that is how time proceeds. So you can see that at the bottom, they were light in color, and that indicates calcium carbonate deposition at the seafloor. And then all of a sudden, they turned brown. And then they slowly recovered again to a lighter color. This change into brown actually indicates that all the calcium carbonate at the seafloor was dissolved at that time, and it was replaced by clay deposition. And so that is an indication that the PETM was associated with a massive input of carbon dioxide. And in addition to that, it was also, so that created dissolution in the ocean, but it also acidified the surface ocean to a degree that we can actually see changes in the ecosystems. So on the right-hand side, you see a figure that shows a sediment core that was located close to ODP 1209 in the Pacific Ocean. And again, you can see the color change from light color to brown color, but you can also see plankton samples from these sediments that have been washed, and you can see they were large before the onset of the PETM. During the PETM in the middle, we can actually see dwarfism of these species. There are even different species in there that occurred only during that event. And then when you look afterwards at the top of that record, you can see that they're larger again, and now they're actually different species than they were before the event. So that is an indication that we had extinction at that time. We had dwarfism, we had changes in the ecosystem. So we wanted to find out how large the acidification actually was at that time. And so we went into ODP 1209 in the Pacific Ocean, and these are results of geochemical analyses that we produce from these planktic foraminifer shells they can see on the right-hand side. Without going into much detail, what you see here is a timeline again. 0 indicates the PETM, or the onset of the PETM of that carbon perturbation, and time goes from right to left. So the part right of it is older than the PETM, and the part left of it is younger than that. On the top panel, you can see carbon isotopes, delta 13C. And the fact that they drop into the PETM and then slowly recover is an indication that we had an input of carbon into the system. The middle panel shows you magnesium-calcium ratios in all three species. And when magnesium-calcium increases, that is an indication for warming. So as we had more carbon dioxide in the system, we also warmed the planet. And this warming is somewhere on the order of about 5 degrees Celsius, or about 9 to 12 degrees Fahrenheit. So it warmed, and then afterwards it recovered, so it dropped down again. On the bottom, you can see boron isotopes. And as I told you before, when boron isotopes decrease, then that is an indication for acidification. And we can see that at the same time, when the carbon isotopes and the magnesium-calcium show changes, we also see a decrease in the boron isotopes. Now, when we try to translate this into seawater pH, we can see that this change in boron isotopes indicates a pH drop on the order of 0.3 pH units. So this is similar to what we're expecting by the end of the century, but again, we have to put this into perspective, and we have to look at how long it actually took to get there. So what happened at the PETM, we had warming on the order of 5 degrees Celsius, pH dropped by about 0.3 pH units. We had ecosystem changes like extinction, dwarfism, changes in the species composition, but it actually took 3,000 to 5,000 years for this event to happen. And so if we calculate what the pH change is per century, then we're still an order of magnitude slower compared to what we're doing today. So again, today was 0.1 to 0.3 pH units per century. At the PETM, it was only 0.01. So that really puts it into perspective, that what we're doing today really outpaces essentially everything that we've done in Earth history. And so that makes it so special. Now I wanted to give you one last event, or one last situation, and that is one that many climate skeptics often use as an indication for carbon dioxide not being such a big problem, or acidification not being such a big problem. And that timescale that we're talking about is the Cretaceous. And during the Cretaceous, we know that it was very, very warm. The carbon dioxide levels were probably as high as 1,000 parts per million, or even higher than that. And yet, we have these chalk deposits that were deposited at that time in the Baltic Sea surrounding England and also Germany and other locations. So massive calcification that happened at a time when the carbon dioxide levels were very high, and obviously the pH of the ocean must have been very low. So what is going on at this time, and why were these organisms able to secrete their shells at that time? The reason for that is that the Cretaceous was a time when the carbon dioxide levels were high for millions of years, for actually tens of millions of years. And what happens on these very long time scales is that the high carbon dioxide levels, they will actually cause weathering of rocks on land, as you can see on this image over here. So weathering of rocks on land actually brings the same elements that organisms want to secrete their shells, the calcium and the bicarbonate ions and carbonate ions, back into the ocean, despite the fact that the pH was low. And so if they have those elements in the ocean, they can calcify, and therefore they could secrete these massive chalk deposits that we can marvel at today. So to bring this to an end, what we know about ocean acidification in Earth history is that it has occurred. It left evidence in the form of interrupted carbonate deposition, reef crises, extinction among marine calcifiers. We know that it goes together with warming and changes in the dissolved oxygen concentrations and nutrients, and in that regard it makes it very, very similar to what we're doing today. And so we can use them as potential analogs, but it turns out that this current rate of carbon dioxide release that we're observing today is, and the oceans respond, that they're really unparalleled in Earth history. And with that, I would like to hand it back to Cassie. Thank you so much for your attention.
Thank you so much, Bärbel. Now we will hear from John Hare.
Great, thank you very much. So yeah, it's remarkable to listen to Bärbel's talk and realize that we're moving into a time period with unprecedented ocean conditions, and that conditions are changing at an unprecedented rate. So it's sobering. So I work for NOAA Fisheries. Our responsibility is providing science for the sustainable management of fisheries resources, and the changes which are occurring present significant challenges to that management. So we're trying to develop tools and information to help managers sort of manage species as the environment is changing into new conditions and changing very rapidly. And as Bärbel indicated, that ocean acidification is not happening by itself. It's part of the larger impacts of climate change, the increased or increasing emissions of CO2 into the atmosphere. And from the northeast U.S., which is where I work, I live and work in Rhode Island, we have evidence. This is a 150-year temperature record from the ocean, and that the line that you see is the long-term change in ocean temperatures. And so it's increased by 1.3 degrees Celsius. We also know that salinity in the ecosystem is changing. Precipitation over land is changing, and stream flow is changing. As Bärbel talked about, ocean acidification is happening. There's evidence for changes in the currents in the region. So the Gulf Stream, which brings warm, salty tropical water up, has been moving northwards. There's changes in wind patterns. The jet stream has shifted northward in recent years. And then there's very good evidence that sea level is rising. And in fact, the northeast U.S. is where some of the sea level rise rates are the highest in the country. So the populations or the resources that we're responsible for providing scientific advice for are experiencing a whole host of changes in their environment. And it's our job working for NOAA Fisheries to try to provide advice to managers as to how to deal with these changes in the environment and the resulting changes in the populations that they manage. And so the other thing that we know from climate modelers is that the climate change issue is going to continue for the foreseeable future. And foreseeable future is kind of a buzzword. It's part of many of the laws that are responsible for resource species in the country. So we know that climate change is going to happen for at least the next 50 years, and then depending on what choices we make, it may continue to happen at a very fast rate, or if we control our CO2 emissions, the rate of climate change may slow in the second half of this century. But this shows projections from climate models of the pH in the ocean in U.S. water. So this is the average projected modeled pH in sort of the 2050 time frame. You can see that there's some variability, higher pHs and lower pHs. But then if you look at the change in pH, so this is pH from the past, pH in the future minus pH in the past, you see that it's uniform. It's uniformly blue. And so these climate models project that pH worldwide will continue to decrease. And again, this blue color is about a 0.1 decrease in pH, but as Bärbel mentioned, pH is a log scale, so that actually represents a 26% decrease in the amount of hydrogen ions in the water. And this is, since the Industrial Revolution began, we've experienced a 0.1 decrease. Climate models suggest that, by the middle of this century, we will have experienced another 0.1 decrease. And then the rate that continues is sort of within our control in terms of our choices about greenhouse gas emissions. And what some of the way climate modelers talk about it is they use these regional concentration pathways, which are basically scenarios about how much CO2 are we going to emit, how is population going to grow, a number of other economic components. And so this is, if you look at the Paris Accord in 2015, sort of the worst case scenario, if we don't sort of deal with what we put on the table in Paris, we'll have a decrease in pH by the middle of the century of 0.1. And so that will create significant challenges for marine resources in the region. So what we've been working on in NOAA Fisheries is developing a tool or a methodology to evaluate the vulnerability of fish and invertebrate species to a changing climate. And this methodology was first used in the Northeast. And it's based on a vulnerability assessment framework. So it's not a numbers model where you parameterize a very specific model for a given population. It's more of a qualitative tool that lets you look at current and existing knowledge and then use expert opinion as to what are the attributes of a given species, what is going to happen in the future from these climate models. And it lets you compare and contrast this quantitative data and qualitative information when data is lacking. And there's a number of steps in this assessment process. I'm not going to go through these now, but I'm going to talk about the results of the assessment in the Northeast U.S. So this assessment takes two components. It has the exposure of species to climate change in a given region, and it has sensitivity. What are the attributes of the different species relative to the ability to respond to climate change? And so in the Northeast, we included 82 species, so we looked into understanding of the sensitivity for all of these different components for all of the different 82 species. And so here you see the ocean acidification factor. If a species is sensitive to an ocean acidification, it gets a higher sensitivity, which contributes to a higher vulnerability. On the exposure side, we looked at the magnitude of sea surface temperature changes projected in the future, the magnitude of air temperature changes, salinity, ocean acidification, precipitation, changes in currents, changes in sea level rise. So here's ocean acidification as part of a number of environmental parameters that are likely to change as a result of climate change. Ocean acidification, about a 0.1 unit by the middle of the century. That goes into the exposure, gets combined with the sensitivity to come up with an overall species vulnerability. So for this exposure of ocean acidification, we use these climate models. And the climate models came from a large international effort called the International Panel of Climate Change Fifth Assessment Report. There are about 13 global models included, and so these global models on average suggest that pH will decrease by about 0.1 by the middle of the century. And so the exposure to ocean acidification, the magnitude of change in ocean acidification is very high. Again, 0.1 doesn't seem like a big number, but it represents a 26% decrease or change in the acidity of the ocean. Then for the sensitivity attributes regarding ocean acidification, we used predominantly an analysis which was sort of summarized all the available information as of 2013. And it looked at different groups of species. So mollusks are the clams and scallops, echinoderms are the sea urchins and starfish, crustaceans are the crabs and lobsters, and fish. And so the study summarized a large number of other studies. And you see that there's a lot of evidence for ocean acidification having a negative impact the survival of clams and scallops, the calcification of clams and scallops, the growth of clams and scallops, the development of clams and scallops. There are some negative effects on echinoderms, sea urchins, and starfish, but there's some areas where the evidence is not clear one way or the other. And then for crustaceans and fish, the evidence is not really clear one way or the other. So in our sort of sensitivity, the mollusks in our 82 species had a high to very high sensitivity. The echinoderms, the sea urchin predominantly, had a moderate to high, and the crustaceans and the fish had a low to moderate sensitivity. And then we combine the biological sensitivity with the climate exposure and end up with a matrix of anticipated vulnerability to climate change in the northeast U.S. region by the middle of the century. And you can see ocean quahog, a type of clam, northern quahog, a type of clam, Atlantic sea scallop, a type of scallop, Atlantic surf clam, a type of clam. These mollusks are showing up as orange, highly vulnerable, or red, very highly vulnerable. And if you look back into the assessment, you see that this vulnerability comes from the impacts of ocean acidification on mollusks. And as Bärbel mentioned, it's the species which form these calcium carbonate shells which seem to be most impacted by ocean acidification. So when we look at the assessment results across the exposure factors, remember we looked at temperature and salinity and ocean acidification and precipitation. We see that in the northeast region, it's really the changes in ocean temperature which had the highest effect, and the changes in ocean acidification which had the highest effect. We also see air temperature as having an effect, and that's going to impact sort of freshwater and shallow water temperatures. So ocean acidification is part of the story in terms of the climate change effects on resource populations, but it's not the whole story. We also have, in this region, changes in temperature which are going to be important. So moving forward, we're taking the results of this assessment and linking it to an assessment of fishing community vulnerability. So our work looked at the effect of climate change on fish and invertebrates. We can then look at the effect of climate change on fish and invertebrates to the communities that depend on catching those fish and invertebrates. So that's sort of trying to get at the human impact of climate change effect on fisheries. The other part, which Hauke will talk about in the next talk, is we looked at sort of ocean acidification at a global scale, but it's more complicated than that. There's regional variability in carbonate chemistry, and this sort of is called coastal acidification. So we need to start bringing in the ideas of coastal acidification into assessments like this. Again, it's not just ocean acidification, it's not just temperature. Climate change represents a whole host of environmental factors that are changing quickly and going to levels which are unprecedented in the geological record. So it's this idea that we need to look at multiple stressors, not just one or the other. From a just a practical point of view, we need to work in terms of doing experiments in conditions that are expected under the next 20 and 30 years. Those are the time scales which are relevant to management. It is very good to know that ocean acidification will have major impacts at the end of the century, but management happens this year, next year, the next 10 years. So we need to start thinking about what the impacts of ocean acidification are on these populations at these shorter 10, 20, 30-year timescales. And then we also need to work on both fisheries and aquaculture species. And these graphs show the value of catch of species in the mid-Atlantic, so that's North Carolina to Connecticut, and in New England, which is Rhode Island to Maine. So this is the mollusks. These are the clams and scallops. So 68% of the value of fisheries in the mid-Atlantic come from mollusks. And those mollusks, if you remember, are the ones which are going to be very highly vulnerable to ocean acidification. If you look in New England, 34% are very highly vulnerable to climate change. So it's critical that we do continue to work and find out more about the effects of ocean acidification on these mollusk species, clams and scallops, and that we also look more at depth and crustaceans. So this 48% of the value in New England is largely lobster. There's been some work, but not much work done on the effects of ocean acidification on lobster. So there's a lot of work to be done, but we hope that our assessment can provide fisheries managers in the Northeast the ability to see where the risks and challenges are that they're going to face in the next, by the middle of the century. This methodology is being applied nationally now. There's assessments underway in the Bering Sea and in the California Current, and in the next couple of years it'll be applied throughout the nation. So with that, I'll stop and pass it over to Hauke.
Thank you, John. To conclude, we will hear from Hauke.
Hi everyone, this is Hauke Kite-Powell. I'm with the Marine Policy Center at the Woods Hole Oceanographic Institution. And we work at the Marine Policy Center on marine resource management issues broadly, including fisheries and aquaculture, and marine environmental topics, including ocean acidification. I want to amplify on some of the things that John and Bärbel have said, focusing in particular on shellfish, because as John mentioned, they are among the most vulnerable and likely to be affected. The reason, of course, is, as Bärbel pointed out, the fact that lower pH in the ocean makes it more difficult for calcifying organisms to build their shells and skeleton, and it particularly affects their survival in early life stages. And this is a problem not just for the ecological integrity of the systems, but for human beings too, who rely on these species for income and for nutrition in many parts of the world. Calcifying shellfish, like oysters, make different forms of calcium carbonate to build their shells. And oysters, for example, build their shells out of a form of calcium carbonate called aragonite early on in their life, and then later switch to making calcite. And it turns out that this is a problem for juvenile early life stage shellfish, because the aragonite form of calcium carbonate is less stable and more vulnerable to dissolution in low pH environments. And this is one of the reasons why larvae and early life stage forms of shellfish are particularly susceptible to these kinds of ocean chemistry changes. John mentioned the concept of coastal acidification, and this is important when thinking about shellfish, because most shellfish farming and harvesting takes place in nearshore coastal waters. And in those waters, the chemistry of seawater is much more dynamic than it is typically in the open ocean. And the reason for that is that biological activity, in particular marine plant life, is heavily concentrated in the coastal oceans. And plants during the day will absorb CO2 from the seawater in the process of photosynthesizing. And when they do that, they decrease the concentration of CO2 in the seawater and thereby raise the pH. At night, when the photosynthesis shuts down, but other kinds of biological respiration continue, CO2 in the coastal waters rises notably, and the pH declines. And so coastal waters generally have a much more variable pH regime than open ocean waters. That variability is amplified in places where people put a lot of additional nutrients into coastal waters, because that amplifies plant growth. In extreme cases, leads to algal blooms and things like that. And that in turn amplifies the cycling of high and low pH conditions over daily and seasonal timescales. And so the coastal acidification story is more complicated than that in the open ocean. And this is relevant particularly to coastal shellfish operations, of course. There are also, as both John and Bärbel mentioned, regional variations in these conditions. For example, the Gulf of Maine waters in New England are particularly acidic naturally already. And this is an issue not just in the nearshore shellfish communities, but also for species like sea scallops, which are wild and on the seabed naturally in conditions often that are already close to the limit of what calcifying organisms like sea scallops can tolerate. And so in some of these locations, small additional changes in ocean pH could have dramatic effects in the coming decades on the viability of those biological stocks. This is a summary of results from lab experiments on the effect of lower pH conditions on a variety of marine species. A paper by Gledhill and colleagues, similar to some of the results that John presented. Again, it's the calcifying organisms, the mollusks in particular, that show the most clear pattern of negative effects. For other species, the effects are less severe or unclear. But an important point also is that people notice differences in subpopulations in the way they respond to lower pH conditions. And that may have to do with the fact that the coastal pH regime is so dynamic, and different subpopulations may adapt and evolve in different ways in response to that. If you look globally at seafood production, very rough numbers, you can see that shellfish in aggregate account for about a quarter or so of the annual value of seafood production around the world. And this is just here a quick note to illustrate the scale of the potential vulnerability of seafood supply over the course of the coming century if conditions really become significantly inhospitable to these animals in large parts of the global ocean. People are looking at global models, as John said, of ocean pH change from the present conditions to what's likely to persist at the end of the century. And for shellfish and other marine organisms, what probably matters is the time, roughly speaking, where the ocean chemistry conditions leave the envelope that these species have been used to living in for the recent historical past. And the time at which that's projected to happen in the models varies from location to location around the globe. But you can do this analysis and see where will change outside of the envelope that local populations of marine organisms have been used to, so to speak. And you can see that these changes will happen in most parts of the world over the course of the coming century, in some places sooner than others. I want to just quickly mention two other assessments that you may find interesting. There was a study published recently by Sarah Cooley and colleagues on the likely effect of acidifying sea conditions on the U.S. scallop fishery. And that includes not just the biogeochemical analysis and the scallop biological submodel, but also a model of the fishery, the socioeconomic component, that attempts to model how the fishery would likely respond to these changes that are coming. It includes estimates of how the scallop growth and survival will be affected by future ocean conditions. There's still some uncertainty about this, but people are doing a lot of experimental work now to help nail this down. And what the model modeling efforts concluded is that certainly by roughly the middle of this coming century, we should expect to see, under the RCP 8.5 emissions scenario that John described before, a significant effect on scallop biomass and on the revenue of the scallop fishery from ocean acidification changes. One other study to mention quickly is one by Ekstrom and colleagues that tries to look at the vulnerability and resilience of fishing communities around the United States in response to these kinds of effects. And so how well a fishing community can respond to or adapt to these sorts of changes depends not just on the biogeochemistry and the biology of the local fish and shellfish stocks, but also the capacity of that community technologically, economically, and socially to adapt to the changes that are coming. And what they find is a high degree of variability, not just in the biogeochemical and physical effects from ocean acidification, but also in the likely ability of fishing communities impacted by this to adapt and respond. And finally, you can look at that on the global scale too. There are big differences in the extent to which countries around the world rely on mollusk harvests for protein or for economic value. And if you combine that sort of information with the likely rate at which conditions will change in the coastal waters of those countries and the economic and technical wherewithal of the fishing populations there to adapt to this, you can assess the vulnerability of different countries to these impacts in the future. So what can we do about this? I want to just highlight 4 possible things that could be done. The first one, and probably a most general one, is we should try hard to limit coastal acidification by reducing the nutrient loading that makes eutrophication and other negative effects happen in coastal waters, and that amplifies the cycles of low pH and low oxygen conditions in coastal waters. Those kinds of steps to limit nutrient inputs to coastal waters can go a long way to making it easier for shellfish species and others there to withstand the changes in broader ocean chemistry. A second possibility, particularly around shellfish farms, is to influence local water chemistry to buffer it, effectively raising the pH around shellfish farms. And one way to do that is to grow large quantities of marine plants in the vicinity of the shellfish farms. Again, these plants absorb carbon dioxide during photosynthesis and effectively raise the pH in the water around them. So people are beginning to experiment with that. Another possibility is to move shellfish hatcheries and farms away from the nearshore waters where the effects are often most traumatic and low pH conditions cause the biggest problems. Some of this has already happened in the Pacific Northwest, where some shellfish growers have moved their hatcheries out of Puget Sound to Hawaii. And a fourth possibility is to select in shellfish farming for populations that prove to be more resilient to low pH and other stresses. So to summarize, I think it's pretty clear now that ocean acidification is a significant potential threat to many marine organisms, particularly calcifying organisms, including mollusks and corals. The effects are often species and location specific, so it's difficult to generalize about the specific things that are likely to happen. We need to think, as John emphasized, about not just pH, but multiple stressors that will accompany climate change, And we need to keep in mind the differences between open ocean conditions and the much more highly variable coastal acidification scenarios. The economic values globally that are potentially at risk from effects of ocean acidification on shellfish are significant, and there are a range of adaptation and mitigation measures that ought to be studied to see how we can respond to this, because even if the world gets serious about carbon emissions, we will be living with ocean acidification effects for the next century at least. And with that, I'm going to hand it back to Cassie.
Thank you so much, Hauke. We're going to go ahead and start taking questions from the audience at this point. The first question, one person asked, I noticed precipitation is not listed as a key factor in biological response to climate change. And this person has also heard that there's been a huge increase in precipitation in the Northeast and that the increase of fresh water flowing into estuaries has caused localized declines in pH. I think this question would probably be best addressed by John. Was this factored into your study, and did you still find no significant impact of precipitation on biological response?
So it's not that there's no significant impact on biological response. It's within the framework of the assessment. The projected future increases in precipitation have more uncertainty in them. So some models suggest, very high precipitation. Some models suggest relatively small increases in precipitation. So as we looked at sort of the exposure to precipitation, because of this uncertainty, the exposure of species was lower. But as the person asking the question rightfully, bases their question on, precipitation can have dramatic impacts on individual species or individual systems. And then it is, gets into the idea of coastal acidification, which in our assessment, we were looking at ocean acidification and not coastal acidification. So I don't mean to give the impression that changes in precipitation, changes in currents, are not important. Rather, the dominant factors in the Northeast appear to be ocean acidification and temperature, but those other factors, sea level rise also, will clearly be very important for some species.
Great, thanks, John. The next couple of questions tie really well into this discussion. The first is sea level rise from thermal expansion and added volume from melting accounted for in pH change projections?
I'm not a climate scientist, so that question would probably be better to an expert with the models. But my understanding of the models which were used in the 5th assessment report is that they predominantly included thermal expansion. So to the extent thermal expansion affects pH, those effects would be included. Ice melt is much more difficult to model, and that's been one of the sort of limitations of the global models that have been used in the 5th assessment report, is the general idea that the sea level rise coming out of those models is a conservative estimate because of the additional sea level rise from ice melt.
Okay. The next question is, how much of the coastal acidification is driven by atmospheric CO2? I know that the modeling was done on general ocean acidification, but if you could address that, that'd be great.
So yeah, coastal acidification presents a significant challenge. Even in the Northeast region, there is a lot of regional variability in carbonate chemistry. So you see differences in pH in the Gulf of Maine compared to the Mid-Atlantic. The global models in the 5th assessment report were relatively coarse resolution, and so they're not really designed to capture sort of the regional scale ocean acidification. So work is going on now to develop sort of regional, they're called downscales. You take a global model, link it to a regional model, and downscale the global effects to your region. And so there's work underway now to generate these sort of downscaled, higher resolution regional models, which will be better able to capture regional and coastal acidification, and then also some of the smaller scale changes in current patterns. But that's work that's going on now, and the results were not available for our assessment when we completed it.
Thank you. In New England waters, how are scientists monitoring ocean and coastal acidification? Are there monitoring instruments that are particularly adept at this?
I guess I'll answer again. Yes, there are. So the monitoring has, both components. There's instruments in coastal regions, and there are also instruments and monitoring activities going on at sea. The number of different instruments. You can measure the CO2 in the water. You can measure pH. You can measure alkalinity, and there's several efforts underway to improve the instruments that are available. So there's a lot going on, and it's a very active area for researchers and for technology development.
Great. I'd like to ask a question of Hauke. Where can states and municipalities go to for ocean acidification information relevant to their region?
I think one place in many cases are the regional ocean observing systems around the United States. So in the Northeast, for example, there's some organization called Narragansett that maintains data sets on marine conditions. Local research institutions are a good resource. The fisheries service offices, like the one that John works in, are another good source. And I think there are NOAA web pages dedicated to ocean acidification. So I think it takes a little bit of searching. There's no single place I would direct everyone to, but those are good places to start.
Great, thank you. I have a question for John. There were fish, particularly flatfish, in the high-risk red boxes with some of the shellfish. Can you elaborate on the life history, diet, or other factors that make some fish species more vulnerable to ocean acidification? And I guess I'll expand this if Hauke wants to comment as well. Are there any factors that make different mollusk species also more vulnerable?
Hauke, do you want to answer first for shellfish, and then I can answer for finfish?
Sure. Yeah, with shellfish, some of the variability in response is due to the extent to which the shellfish rely on aragonite versus calcite to build their shells at different points in their life cycle. Different species of shellfish use different biochemical techniques for building their skeletons, another source of variability. And I think that when it comes to other marine species, there's some evidence that acidification affects the way that squid, for example, and I think some finfish as well, are able to build their internal organs, including those responsible for allowing them to navigate and find their way in the water column. And so there's a lot of research on this now, and a lot that we still don't know about it. But I think that in the coming 5 to 10 years, we'll get a much better fix on exactly which species are likely to be affected by this. And John, you may be able to add more there.
Yeah, just a little bit. I agree that the next 5 to 10 years, we will know a lot more about the effects of ocean acidification on finfish, on shellfish, on mollusks, on the crabs and lobsters, and that information will be critical to understand how ocean acidification is going to affect the resource species. From the fish side, as Hauke mentioned, there is this sort of several studies which indicate that ocean acidification may affect fish's ability to smell, And so it's, the mechanisms aren't exactly clear, but, the change in carbonate chemistry is just, it's having a wide range of effects across multiple groups of organisms, affecting calcification, potentially affecting olfaction, affecting physiology. So we're really just beginning to learn what the impacts are going to be.
Thanks. I have a question that I'm going to address to Bärbel. So far as you can tell, because I know it's very difficult to estimate pH through geologic history, but has global ocean pH ever been as low as the projected RCP 8.5, which is approximately pH of 7.5?
7.5 is very low. It's probable that during the Cretaceous it was that low as it's going to be. But as I tried to explain before, the difference between the Cretaceous and today is that the carbon dioxide levels were high for a very long time. So for this weathering of rocks on land, for that effect to kick in, that takes about 20 to 40,000 years. And so I don't know whether you remember that in my figure. The recovery at the PETM actually took somewhere on the order of 70 to 100,000 years from that acidification condition. So that kind of puts it into perspective of how long this is not an effect like the ozone hole or acid rain that we can easily address and reverse. It is something that will stick with us for a very long time. So even though we have seen these low values, they have not actually been put into place at a rate as we are putting it into place today. And that makes the big difference.
Great. So I think this will be the last question. In some reef fish, the adult and young of the first generation exposed are greatly affected by ocean acidification. However, some second generation fish are not affected. Is any work like this being done for higher latitude fish species?
Yes, I know of several projects where this is being done, and this raises an important issue in general about how we think about the effects of climate change on marine species. Just to make the situations more tractable, we oftentimes make the assumption that the biology or ecology of the species is not going to change going forward. But the person who asked the question, identified studies which show that individuals can acclimate to new conditions. So, as when presented by new conditions, their physiology can make changes so that they're better suited for those new conditions. And groups of individuals, populations, can also adapt to new conditions. That's an evolutionary response, and that can happen relatively quickly too. And we don't really have a good idea of what the acclimation capacity is or what the adaptive capacity is for most marine species to ocean acidification, to warming temperature, to changes in stream flow, and that is an area of active research.
Great. Thank you. That's all of our questions for today's webinar. If you have any questions that were not addressed today, you can email them to us at cipinfo@americangeosciences.org, and we'll make sure to send your questions along to the speakers. We will be posting today's recorded webinar on our Critical Issues webinar page soon. You can visit the website to view this webinar as well as previous ones and check out the webinar schedule for upcoming webinars. And again, we have another webinar coming up on March 30th. Thank you very much, Bärbel, John, and Hauke for speaking today. This concludes our webinar for today. Thank you for joining us, and we look forward to seeing you at our next webinar.